Thank you, Jack.
A couple of weeks ago, there was a paper looking at lithium in cryo-EM, so even lower Z materials. Is there any talk about that at Alamos?
Yeah, I don't really know. Sorry.
Just wondering.
Okay. Oh, yeah.
Can you look at the ribosome with the messenger RNA in it?
Yeah. The question is, can you look at the ribosome with the messenger RNA in it? And the answer is yes, you can. It's a good question. They usually have to have special sequences that sort of are basically when you try to image these ribosomes, they try to trap it with different drugs, basically. So if it's stopped, it's easier to image. And then also with the messenger RNA, if they have a special sequence, it's easier to image because it's stopped. Yeah.
Can you put something in, like a sequence of DNA that stops at a particular point?
Yes, you can. Yep. Mm-hmm.
Repeat the question.
Oh, sorry. Can you put something in like a sequence of DNA that can stop the ribosome at a particular point? Yes, you can put in a sequence like that. So there's one in the back.
Hi, my background's in biology. I was wondering if you need to have every single one of those hundreds of thousands of atoms in order to create that same function at lysosome, or if there's any flexibility in the structure, or maybe.
Yeah, this is a good question. So the question is, basically, for this case of the ribosome, if we have looking at 1 million of them, do they all have to be exactly the same? The short answer is yes. And so if they're all the same, then you can see every atom. We can't quite see every atom now. Every atom would be like 1 angstrom resolution, and now they're around 2.5 angstrom. And the 2.5 angstrom is because they're in slightly different configurations. And so what they try to do are play tricks, like putting in different drug molecules and stuff so that they can get all of them in the identical state. But even then, there are pieces of the ribosome that are intrinsically flexible. So for this case, there's certain regions that we'll probably never see with atomic resolution. Yeah.
So in another imaging system, you've got like synchrotron where everything in the laser is lined up. Would something lining up the phases of the electrons help in this case, or be detrimental? Or?
I don't know the answer, but a related answer is I know one thing they're doing is another issue is that it turns out for cryo-EM that if it's a big huge thing, it's easier because you actually you get these images from the microscope, and in the old days the graduate students had to look through and pick out the 1 million ribosomes like one by one from the noise, and if they're bigger they're easier to pick out. Now people are pushing the envelope to look at small things, and what they're doing is they're lining all the proteins up by having them glom onto special sequences of DNA. So they make like this DNA lattice, and then they have all the proteins identically aligned, and then they can image even small things like a single protein. Yeah.
Is it possible to modify ribosomes to the point that you produce synthetic material?
That's a great question. That's kind of like is it possible to make a ribosome that could create like a synthetic material? And that's kind of a holy grail for like nanotech or biotech, I think, because then you could kind of make because these biomolecules are basically like programmable material, and then if you could harness that would be really useful. And so people have they have engineered these. Well, basically proteins are made out of amino acid building blocks, and they've engineered the ribosome to introduce artificial amino acids into proteins. So they're making steps in that direction. Okay, or one more? Okay.
So I noticed in the ribosome it looks like it has kind of a double helix structure, at least in parts of it. Is it structurally similar to DNA?
Yeah, so the question is the ribosome structurally similar to DNA? And the answer is yes, it is. So basically it's sort of like a big piece of DNA that's wound up in a ball, like a bowl of spaghetti, pretty much. And it turns out that RNA is ribonucleic acid and DNA is deoxyribonucleic acid. There's basically only one chemical group difference, 2' hydroxyl on the backbone. And that allows like these weird configurations to happen in RNA but not DNA. But it's basically like double helices everywhere, but it's wound up like a bowl of spaghetti, pretty much. Yeah. Yeah.
So someone asked the question earlier about different ribosomes, and so now I'm confused because I thought like the human ribosome was the human ribosome.
Right.
And it was the same ribosome in whatever cell is in your body, making whatever protein. Am I misunderstanding
Okay,
this?
the question is, are all ribosomes the same? Is that,
At least human ribosome.
are all human ribosomes the same? Okay, this is really a cutting edge area of research right now, and there's like a paper that came out like a week ago, I think, where they have imaged the human ribosome, and it turns out that like in DNA you have 4 bases. You have A, G, C, and T. In RNA you have a U instead of the T, so it's A, G, C, and U. But it turns out there's a whole zoology of sort of customizations of those bases, modifications to those bases. And in the field of epigenetics, those modifications are critical in determining how embryos develop and everything. They control all the switching. And so people have speculated that maybe that happens on the ribosome, and then this paper that came out last week says that yes, it does. And like, so they were able to image all the little modifications all around the ribosome, and they're all over the place. So what people think is that the ribosomes in different cell types are slightly different, and they have different modifications to their RNA that may allow them to talk to other things in the cell differently. And so like a cancer ribosome might be different than a non-cancer ribosome.
So if you had the choice of which ribosome specifically to work on next to understand its dynamics, which one would it be?
Yeah,
knowing this.
my friend in Cornell is doing cancer, kidney cancer ribosomes right now, I think. So that's one interesting area, yeah. And so the ribosomes are responsible for making all the protein, all the substance of your cells. So in cancer, you have uncontrolled growth, and so you sort of have uncontrolled ribosomes in cancer. And so, but they've only been able to image the human ribosome a few years ago. So it's so new that people haven't, that we're just starting to look at this right now.
Are there other applications for this that now that they've got it and it's coming up, that they're going to start applying it to?
Yeah, there are many other applications, and basically up until 2012, in 2012 the first Alamos structure of the yeast non-bacterial ribosome was solved, and then human came a few years after that. We had one of the first human structures. But most of the applications are non-bacteria, so basically all the applications are like fair game now. So there are many cancers, all kinds of different kinds of human disease that will benefit from this kind of research. So.
Are there ways to change the ribosomes?
Are there ways to change the ribosomes? Yes, this is how we do most of our research. So the ribosome is mostly RNA, but it has 50 proteins stuck inside it all over the place that kind of glue it together. You can knock out any of those proteins, you can mutate the proteins, you can mutate the RNA. There are all kinds of things you can do. Yeah.
So besides looking at that actual configuration of the ribosome, you're specifically looking at also the motion of the ribosome and exactly how it moves to work as a nanoprinter of proteins, if you will. What is your next step to go forward in your research?
Okay, I will grab this. So we're focused. The ribosome is really complicated. You know, basically it has these 5,000 bases on it. And it looks like a big ball of spaghetti, but it's identical